Ira, and I'm from Edagene, a biotech company dedicated to advancing genome engineering with innovative CRISPR-based technologies. Today, I'm going to walk you through one of our most exciting technical breakthroughs, Flash KI, our next-generation CRISPR knock-in technology. So in this video, I'll introduce you to why gene knock-in matters in research and cell therapy, the weaknesses of conventional knock-in systems, how Flash KI overcomes these limitations, and real experimental data and performance results. So let's get started. Gene knock-in technologies play a crucial role in basic research and cell therapy. However, conventional knock-in methods, typically using plasmids, electroporation, or RNP combined with AAV, universally face three major challenges. First, conventional systems often rely on electroporation to co-deliver sgRNA and donor plasmid into cells. But plasmid systems depend on transcription inside the nucleus, which causes slow, delayed, and unpredictable editing component expressions and overall low efficiency. Then there is the RNP plus the AAV method. Although AAV increases the DNA cleavage efficiency, AAV delivery still suffers from the following: packaging size limits, potential immunogenicity, and restrictive delivery conditions across different cell types. So no matter whether it is physical or chemical delivery, the knock-in efficiencies in most mammalian cells remains very low. Second, electroporation produces significant membrane disruption and cell stress, and lipid-based transfection causes serious toxicity as well. These delivery methods severely reduce cell viability and compromise downstream applications. Third, homology-directed repair has inherently low efficiency because it is only active during the S and G2 phases of the cell cycle, and it requires a homologous donor repair template to achieve precise editing. In addition, mammalian cells naturally favor the non-homologous end joining pathway, which rapidly seals double-strand breaks but does so in an error-prone manner over HDR. These biological constraints make traditional knock-in strategies difficult, inconsistent, and highly limited for research and therapeutic applications. To overcome the fundamental limitations of traditional knock-in systems, Edagene developed Flash KI, a next-generation knock-in platform designed to improve delivery efficiency, minimize cellular toxicity, and enhance HDR-mediated repair. Flash KI combines two core innovations: a high-efficiency delivery strategy known as flash packaging and a specialized KI enhancer drug, which work together to make knock-in significantly more effective and more predictable. Flash packaging enables CRISPR RNP and donor DNA to be co-capsulated into a safe and highly efficient delivery vector. This vector then transports the complete editing machinery directly into the nucleus without electroporation or lipid-based reagents, ensuring gentle yet high-efficiency delivery. As a result, Flash KI dramatically reduces the cellular stress and toxicity associated with conventional knock-in approaches, while also performing exceptionally well in difficult-to-transfect cell types. Alongside the optimized delivery system, Flash KI incorporates a KI enhancer drug that fine-tunes the intracellular environment during a critical editing window. The KI enhancer suppresses the competing NHEJ pathway and simultaneously promotes HDR-mediated repair, which maximizes precise insertions and greatly improves knock-in efficiency. In another word, Flash KI solves both the delivery problem and the HDR problem at the same time. With these two components working synergistically, the Flash KI workflow becomes remarkably simple. Delivery and editing are completed within 24 hours, and knock-in efficiency reaches peak level within 7 days. This rapid and efficient process enables the reliable generation of healthy, high-percentage knock-in cell population without the need for drug selection or labor-intensive enrichment steps. So, overall, Flash KI solves the three major limitations of traditional knock-ins by having higher delivery efficiency, minimizes the cellular damage, and having a much higher HDR performance. To see this in action, here we can see a direct comparison between traditional electroporation and the Flash KI system in HEC293 T cells. Both methods use the same CRISPR RNP and the same EGFP donor design, targeting the C-terminus of the GAPDH gene. Under electroporation, we achieved about 68% knock-in efficiency, which is considered quite high for this cell line, but at the cost of noticeable stress and cytotoxicity. With Flash KI, however, the knock-in efficiency increased to about 88%, and the cells maintained a much healthier morphology. This highlights a key point for us. Flash KI is not only more efficient, but also significantly gentler on cells compared to electroporation. But now the key question is, how does this translate into real experimental performance? To evaluate this, we tested EGFP knock-in in A9 cells, a cell type known for its extremely low baseline HDR activity. When Flash KI was used without the KI enhancer, the knock-in efficiency remained at only 0.2%. which aligned with the typical sub% efficiency seen in difficult-to-edit cell lines. However, when the KI enhancer was added, the performance changed dramatically. Flash KI increased the knock-in efficiency from 0.2% to 46%, which represents an exact 230-fold improvement. This dramatic increase reflects the synergy between Flash KI's high-efficiency delivery system and the KI enhancer's ability to shift the repair pathway toward HDR. And importantly, the Flash KI-treated cells also maintain healthy morphology and viability, showing that the system achieves high efficiency without compromising cell health. So, in summary, Flash KI offers high-efficiency delivery, very low cytotoxicity, strong HDR enhancement, faster workflow and higher knock-in percentages, and excellent universality across mammalian cells. At Edagene, our mission is to empower the scientific community with reliable and innovative gene editing solutions that push the limits of what's possible. Now, thank you for watching, and we look forward to supporting your next knock-in project with our Flash KI.